Thank you, Nir. Thank you to the conference organizers. I'm really delighted to be here to tell you about how we came up with and what our concept is behind the TAME trial. I will observe that I came into this process quite skeptical that such a trial could be designed, actually, and I was very, this has happened to me before. When you start digging in, you start seeing what the problems actually are, and it turned out to be much more tractable than I had anticipated. So I'll tell you about that a little bit. There we go. So you just heard about the interventions testing program from Nir. This is a step in the translational pipeline to look at good targets to move forward in aging. And then you start realizing that a mouse isn't a little human, a short-lived human. Most of the interventions testing programs, interventions, either start giving the mice the test substance either at 4 months of age or 12 months of age. And if you think about what that means in terms of the proportion of human lifespan, that's somewhere between starting at 18 and 40 years old. And then this program follows them until all the mice die. So if you translate into human terms, you're realizing that's a 40-year-long study of testing something that we don't know if it's even going to work. So we use this kind of information for leads, but that paradigm isn't really going to work very well as we move into humans. So there is a continuum of evaluation that goes from all of which we can do in humans, starting with looking at changes in gene expression to see if the right genes are up or down regulated according to the clues from the animal models. We can look at changes in cellular physiology to see if those are consistent with what we understand about the biology of aging. We can look at changes in mortality-associated biomarkers. All of these can be changed in a relatively short period of time. This might be days and weeks. This might be months. Then there's another set of markers. We can look to slow the degeneration across organ systems. We can't make them better, but we can make them worse, slow down the rate at which they decline. Things like kidney function and lung function fall into this category. We can retard the emergence of age-related disease, lower mortality rate, and extend lifespan. We're not interested in this. We're really interested in this middle area as a way to increase health span in people. But as you can see, as we move through this continuum, time increases, its expense, but also the salience. Who cares about the answer? But we really care about this. People will not be all that excited. They'll be interested, but this is mostly for the scientists and not for the general public. Now, in our discussions with FDA, they're really only interested in this part of the space at this point in time. If you had a new antihypertensive drug, you're back here because there's a wealth of information linking the change in blood pressure to lower stroke, lower cardiovascular disease. But if you're trying to target aging, it's not clear what these markers actually ought to be. We have a lot of epidemiology to suggest it, but it's not entirely clear. Where they can't, in our discussions with FDA, it was clear that if you could delay clinically manifest what sometimes called hard endpoints, this is something they'd be interested in. This is the kind of information they need to develop an indication. Changes in biomarkers is not a disease, it's a change in a physiologic measure. And at this stage in the evolution of this idea, they're not willing to consider this as sufficient evidence for an indication for a drug. So because of those interactions, we're in this part of the continuum of evaluation. So if you're going to design a randomized clinical trial, and we use the randomized clinical trial because FDA agrees that that is the most rigorous approach for evaluating or establishing the efficacy of a treatment, a potential treatment for a potential target, you've got a few things you have to design or decide. What treatment, what outcomes, how frequent is the outcome if you don't give any treatment, and how big of an effect are you planning to find? And this will tell you really the key benchmarks that you need to move a trial forward. And so let's tell you about that. So you've heard a lot about metformin in this meeting, so I don't have to go through that. It's very lots of clinical experience with this, and I'll talk about safety in a second. Here's some additional information. Metformin started at midlife, increases lifespan and health span in mice. So this is this report from well, I know you can't read the citation in the audience because I know I couldn't read them, but it's from a 2013 publication. But more importantly, health span improved. Although a short 6% extension of lifespan, a 25% health span benefit. And you can look at all the variety of readouts, both at the biologic level, the functional level, the disease level, all improving with the administration of metformin in this mouse model. What also makes metformin attractive, you've heard from Nir how it targets the biology of aging, but we're trying to prevent things in people who don't necessarily have anything. So safety becomes the preeminent consideration in a prevention trial, because we don't know we're doing anyone any favors, so at least we shouldn't do them any harm. And metformin has an excellent safety track record that is very important in the risk-benefit balance as you're proposing a trial to go ahead with. And very practically, since it's a generic, it's very inexpensive, and it's pennies a day to actually do this. If you're in a big trial, any number times pennies a day is still a big number, but at least it's a tractable one. So what is the outcome for a clinical trial targeting aging that satisfies the FDA's desire to have hard endpoints as the focus? Well, we ended up discussing this, and we decided that by the hypothesis of targeting aging, if the drug is effective in that targeting, you would reduce the occurrence of multiple diseases which mostly have the common risk factor of aging and maybe not a lot of other common risk factors. And this collection is a composite endpoint. These are quite common, where it's not one disease, but a composite of several diseases. And the outcome of interest is then the time until the occurrence of any one of the diseases that are part of the composite. So the components of the composite should be age-related, common, and pose a significant health burden, and cut across major disease families. This third one is especially important if you're say you're targeting aging. So these are data from the Rochester Epidemiology Project in Rochester, Minnesota. Our Department of Health and Human Services has identified 20 diseases that they use to come up with their comorbidity burden index. And I'll just point out that you can see some of these are quite clearly age-related in a very dramatic way. Some substance abuse, asthma, not so much, but most of the diseases people worry about, these contribute to most of the healthcare expenditure in the United States are either from these 12 or from these remaining 8. And many of these, so these are all candidate to put into the basket for your composite outcome. You should also have some evidence that the interventions targeting aging might affect these diseases. This is where the animal models are very important. Is cancer slowed? Is vascular disease? Is Alzheimer's slowed? drugs targeting aging do slow all those processes in animal models. And because we're trying to figure out how important metformin might be, it's really good if there's some experience about the effect of metformin on those disease processes in humans, so we know what we're looking for. So first, let's talk about metformin and all-cause mortality. So these are epidemiologic data, observational data, so they don't prove that metformin has an effect on mortality. If it didn't have an effect on mortality, you wouldn't see one in the epidemiologic studies. So not seeing an association rules out anything, doesn't necessarily rule it in. That's why we do clinical trials. The epi gives us leads to follow up in clinical trials. And across these 4 studies, you can see a 7% overall reduction in total mortality across in this meta-analysis of 4 studies. This is very important. This is from the UK PDS study, which gave new onset people with newly diagnosed diabetes, either standard of care or metformin. And you can see about a 25% reduction in total mortality in those people randomized to the metformin arm. So compared to other kinds of treatment in diabetes in a randomized sense, there is a reduction in total mortality, and in observational studies, also primarily exclusively in people with diabetes, type 2 diabetes, you see also a decrease in mortality risk. There are quite a few observational studies on CVD outcomes, and you can see over here the expected reductions in cardiovascular disease, 21%, 19%, depending on the subgroup, 19 to 32%, 24% reduction. So very consistent evidence from epidemiologic studies, which is also consistent with randomized clinical trials in people with diabetes of a substantial reduction in cardiovascular disease. Most of the cancer data you can use is from observational studies, and so this is reduction in cancer incidence that's observed in people randomized to metformin, or not randomized, taking metformin as opposed to other agents, and a 31% reduction in cancer incidence and a 33% reduction in cancer mortality from this meta-analysis. The data on cognition is emerging. It's not been a major focus, but we've realized that all the major risk factors for dementia that aren't ApoE4 all look like cardiometabolic disease. And so there's a lot of interest at this confluence of metabolism and cognition. So the literature is a little bit inconsistent, and the quality of the studies mostly isn't very good. This very recent study looking at US veterans from our Veterans Administration who have diabetes was quite well done and found at least an observational substantial reduction in the risk of dementia. And when they do very fancy statistical weighting of the folks to try to mimic what a clinical trial would observe, they still see a reduction in risk, particularly at the 65 to 75-year-old age group. So that's the basket then we're proposing for the TAME trial, which is cardiovascular disease and heart failure, cancer, MCI, dementia, and death. We have some other sensitivity analyses, but I can talk about them if there's questions about those. So the design of the study, briefly, is a double-blind randomized clinical placebo-controlled trial. It will have a 2-week run-in where everyone gets a low dose of metformin, and people can't tolerate it. They will be excused from the study and then randomized to either placebo or an escalating dose of metformin to get up to 750 milligrams twice a day. Medium follow-up time we're planning is 45 months. The exclusions are diabetes. It's not ethical to randomize people to a drug to treat diabetes if they have diabetes. So these are people without diabetes. without impaired kidney function because of the labeling. They don't have dementia or other life-threatening illness. And this is how it looks. I'm going to talk about the gait speed in just a second. We're targeting people 65 to 80 with gait speed at less than 1 meter per second. This is to develop inclusion criteria that allow enough events to occur for us to have statistical power to answer the question that we want to answer. We're targeting 3,000 people. We've already talked about the primary endpoint. But this trial has a lot of audiences. So the gerontologists and geriatricians are interested in a composite functional outcome of decline in mobility, cognitive function, or death. And we're going to look also at the rate of accumulation of all those 14 age-related comorbidities that I showed you in the earlier slide. This is really a great test. If you can take this agnostic basket of age-related comorbidities and slow their accumulation through time, you have very strong evidence that you've targeted aging and not looked at a specific pathway. So how many people, how do we come up with the 3,000? Well, this gives you, we have many data sets that we model the inclusion criteria to see how fast the endpoint is achieved. This is a trial of 1,600 people that we looked at our inclusion criteria. These people were recruited to be somewhat physically impaired. Over the 5 years that we're proposing, 42% of them would hit endpoint at the end of 5 years. We have another population, the Health ABC study. These are well-functioning community-dwelling adults. If we apply these entry criteria, about 35% of them develop the endpoint after 5 years. If we used people with higher gait speed, this is much lower. So just knowing their gait speed tells you a lot about the future health of folks. We're planning for an event rate of 7.5% because it's everyone's experience that everyone's planning is always very optimistic with respect to what you actually see when you actually put people in the trial. So then we're hoping to deflect this curve down between 20 and 25%. And so this is our effect size, which came from reviewing of all the previous clinical trial data and all the epidemiologic evidence. And we think we have 80% power. We can talk about what that means if people have questions for a 20% deflection and over 95% power for a 25%, which translates into about a 20% accumulation in this group over 5 years versus the expected accumulation of about 26% in the placebo group. So this is really a schematic that one of my graphically talented colleagues, Jamie Justice, put together. She did many of the prettier slides or things that she put together. So summary points. I didn't think this trial was feasible. It is. It's not any bigger than many prevention trials. It's smaller than most, like the Women's Health Initiative or Mr. Fit. There's no single correct endpoint. Different kinds of endpoints speak to different audiences, and the basket of hard clinical diseases speaks to the FDA audience. And this is a building block because you need a trial with the clear-cut endpoints to validate biomarkers that can be used to accelerate the pace of aging research by knowing which ones changed were associated with how much disease reduction. So thank you very much. I'm from Winston-Salem, which does not have an international reputation, so there we are. It's a lovely place, so thank you.
We're actually doing pretty well on time, so as the moderator, I'll take the prerogative of asking a question.
Sure.
Gait speed. Is that something that's been used in other clinical trials? You know, because the more traditional endpoint is 6-minute walk, which has actually been used for some approvals in the past.
Right. So this is an interesting point. The gait speed is not an endpoint. It is an entry criteria. And the people who study aging have come to embrace gait speed. Every 0.1 meters per second difference in gait speed is associated with a 12% difference in overall mortality risk for older people. It predicts hospitalizations, it predicts disability, it predicts all-cause mortality, obviously. So, and it's quite clearly aging related, because your gait speed is pretty constant through most of your life, and then as you hit this age, it starts declining, and people who decline faster in their gait speed are at higher risk of mortality. And so it is a very integrative measure, and you can use it to stratify populations to identify people who are at high risk for developing a variety of diseases. So it's a part of our toolkit, and it works quite well when we model it in populations.
Can I add another important point?
Oh, sorry. We got the microphone here.
One of the problems with studies is that you get more healthy volunteers. We're very worried about metformin in particular, because there's lots of people who want to come to the study, and they wouldn't know this criteria, but if we get people and we have objective way to fail them, we'll get less of those people, or the one who will become centenarians anyhow, right? We want to avoid that. So it's both.
So you selected metformin dose 750 milligrams BID.
Yes.
The entry-level dose is 500 milligrams BID. These are subjects without diabetes. What was the basis of choosing that? Because you're likely going to experience a higher rate of adverse events compared to 500 milligrams BID.
Well, we based that on the experience with the DPP, which was the dose that they used in people without diabetes. And we expect some 10 to 12% will fail the run-in, and then people would be escalated, but if they got intolerant at higher doses, they would stay at whatever dose they could, the dose that they could tolerate up to that point. And I don't know, Nir might have some additional thoughts on the selection of the dose.
Maybe I'll come with
Yeah, why don't you? That's probably more efficient.
Just to add to those of you who don't know, but metformin is not a hypoglycemic drug. It's an anti-hyperglycemic drug. It improves insulin action or cellular function. So you give it to people, they're not going to become hypoglycemic. And the DPP has experience, but there's experience all over from clinical and other studies that you give it to normal people. Each one of you can take metformin. You're not going to be hypoglycemic unless you lose a lot of weight sometimes, but that's a different issue. So I don't know if you had any other, when you said adverse effect, I'm not sure what other things you had in.
GI.
Oh, GI. Well, that's why we do run-in period for 2 weeks. And well, you usually see it in the first week and it goes away. If it doesn't go away, we'll exclude.
The man with the microphone.
I would like to make a point on setting up this type of studies where you introduce a big basket of phenomena, basically, that you want to study. Basically, the risk is dilution of your endpoints and dilution of the effects. Has it been considered, if we think about common pathways, to enrich this study with a metabolomics type of exercise to see whether when you go along with this study, which basically is a hallmark type of study, you should, I think, take the opportunity to work on biomarkers that could give the results a bit more easier. And there is a metabolomics initiative in Leiden, has started with a lot of pharmaceutical companies trying to establish baseline metabolomic profiles, but that's in younger people currently. It would be good to extend that to elderly people, and then maybe you can use this type of results to introduce in these studies that you're talking about now.
So that's a great comment, and we have a very extensive biobanking part of this initiative to provide raw material for people to do exactly what you suggest. So thank you.
I'd like to ask the question about the study population.
Yes.
Prior to randomization, is that study population, is this a copy of the total population from socioeconomic status, educational level, race, etc.? Or have you chosen this population out of a specific part of the total population that you had? I'm asking the question because of the fact that if, for example, you do not include people of different races and people of low socioeconomic status, what impact has this potentially got on the relevance of the findings that you have?
Right. So generalizability is definitely a concern of ours. We plan to recruit 50% men and 50% women, which is the first. We plan to recruit in proportion to the ethnic minorities in the study centers, and there are 14 study centers in this, I think. With respect to, and so the criteria that I laid out for you, low gait speed, no diabetes, appropriate renal function, is about 40% of the people in this age range would qualify. Recruiting people of low SES is a real challenge, at least for us in Winston-Salem. The people who are engaged in this sort of thing tend to be from higher SES, so that's something we work on. But other of the centers among the many participating have different SES makeups in the people that they attract. But it's a point well taken. Yes, sir. He's been waiting over here. So.
Yeah, I have a similar generalizability question, and I agree as a trialist, this is a really good population to study because you're going to have the endpoints that you want to have in the population, but I would guess that fewer than 1 out of 5 70-year-olds is going to qualify for this study for a variety of reasons. So is it going to really be generalizable to the relatively healthy, as you said, you like to intervene early, 65, 70 to 75-year-old population?
Well, first you need to understand, people can come into the study with these diseases. So they're not a clean, non-disease population. If we were recruiting only people who had none of those manifestations, I would agree. That would be a very unrepresentative group.
We're going to prevent the next disease, not the past one, right?
I'm looking at it from the other perspective. Yeah. If you exclude the relatively healthy 70-year-old, which you are, very few people.
Yes, right.
How are the results going to be generalizable to the relatively healthy 70-year-old?
Well, this is the constant tension of all clinical efficacy studies, because the FDA, the NIH funds you for 5 years. So you need to figure out something that you can do in that time period. And so in that time period, you need to recruit a population that will have events at a fast enough rate in order for you to hit your sample size. And then usually what happens is once the efficacy is established in any population, then there's interest in expanding the boundaries of that finding up and down and sideways. But it is a tension that is really unresolvable. This was true for the first drug to prevent heart disease. It was only given to men with type 2B hyperlipidemia, which represented 5% worse people, highest LDL levels, and only men. That was the LRC-CPPT. So was that the only study ever done? No, there were lots after that expanded the range of people in whom hyperlipidemia ought to be treated.
And I'll point out that this study wouldn't answer the question of when you should start being on metformin, right? Because we want to get it fast and we want to get it, through the FDA. So that's another problem. What are you telling people? Should you start it when you're 40 or 50? We won't be able.
Right. So since I'm the next speaker, I'm going to actually yield 2 minutes of my time to the gentleman from Newcastle, and then I'm going to have Zan ask the last question. Okay. But we have a question here.
Thank you. it's really a point which makes it quicker, doesn't it? First of all, the pharmacoepidemiological studies of metformin are junk. And you have to realize that they're confounded by the contraindications which medics regard metformin of having in liver disease, heart disease, and kidney disease, and particularly those related to SU due to the stepped pathway and where metformin is used in diabetes. So don't be misled by them. They're a mess. Secondly, and importantly, as clinical trialists, the devil is in the detail here. The FDA has recently refused a huge CVOT to get into the label of one drug from one major manufacturer. And we don't know why that is, of course, because the complete response letter is confidential, but we can guess it's because of uncertainty of how the outcomes were defined and whether they're rigorous. And you have probably, I guess, got quite a major problem in that area because you've got so many outcomes.
Yeah, well, let me address the second, and you will address the first. A lot of our budget for this study is on outcomes, ascertainment, and surveillance. So that's a very well taken point. You're on the first point.
Yeah, I actually, let me make this point in your words. Don't be misled. I don't want you to mislead here anyone. What happened is when metformin came up, it had a lot of black labels related to congestive heart failure, kidney disease, and liver disease. In an Annals review last year, and then the labels kind of got loosened up because metformin was so good. In a recent Annals paper, it was shown that the people with congestive heart failure, kidney failure, and liver failure that got metformin had a decrease of mortality between 25 and 55%. In other words, because nobody knew that metformin targets aging and that those diseases are hallmark of aging many times, they were prevented in those people who should use that. So let's understand, it targets aging, and those people are doing much better on metformin, and that's important to note.
But also to your point, that's why we do the clinical trial, is we have this stuff and we don't quite know what to make of it, and so this is the only way to get an answer we can depend on. So, yeah.
Well, Nir and Steve, this is such a vitally important trial. It's very thoughtfully conceived. I like the idea that you're using gait speed to select patients. You're not using it as an endpoint, as you said. It's worth noting, though, that gait speed is probably the likely candidate for sarcopenia outcome trials. Yeah. Best functional practical measure for sarcopenia trials. You might ask, well, I'm sure you're going to look at the effect on gait speed. But it's a good way of enriching the population. And that's, I think, the point that Art Santoro was bringing up, that you're not, you don't have a representative population of elderly people, but this is very analogous to the cardiovascular safety trials we've been doing for diabetes drugs, where we don't treat normal people. We take people almost out of the CCU who've just had an MI. We don't know if the results are really generalizable to the general population. They probably aren't, but it's a way that you get an event rate that is sufficient to power your study. So it makes perfect sense. It's a way to get started. It won't be the definitive demonstration, as you say. You'll need to take a next step. My only concern is that in our experience with cardiovascular safety trials, mortality has typically not contributed to the composite. It's usually been MI, stroke, but mainly MI. So there is a concern that, ironically, even though you're going to have certainly a very high mortality event rate, it may nonetheless be too much for metformin to move the needle on mortality. That would be my only concern about that composite.
So that's a point that people pick whichever part of the basket, and they all make that same point about each individual part of the basket, which is the point of the basket, actually. If we move the needle a little bit on each component, we're not powered for any particular component of the basket. We're powered for the whole basket. And so, yes, you're right, we might not move the needle a lot on mortality, but if we move the needle a little bit on all the components, that's actually more persuasive evidence that we've affected the aging process than if we had a huge effect on cardiovascular disease and no effect on anything else, and then we had a very nice cardiovascular disease study, but really haven't advanced the cause of targeting aging.
You're wrong, of course. You're showing is the effect on the positive,
Yes.
not the effect on aging.
Right.
That's very important.
Yeah, no, I think you're right, but the logic is that if you did affect aging, you would affect the basket, and the basket is something the FDA might give an indication for, and aging, they've told us quite explicitly, is not. And so until we cut that Gordian knot there, the whole field is stuck. So.
Well, and as I start my talk, I'd love to know, what division do you go to for aging? Because, of course, the way FDA is set up, it's by disease. So there is no aging division.
Well, so by the way, Bob Temple is adopting us, right? So he's...
Well, because, of course, he's aging by the minute.
Right. And actually, when we went to the FDA, Ron Howard, the producer, was following us with camera. We went to the Senate, to the FDA. He did a breakthrough episode about the age of aging because he thinks next decade we'll start targeting aging very well. And he couldn't go to the meeting, but he had an interview with Bob Temple. What Bob Temple said in the meeting, he said, I don't buy necessary aging. I don't know if you do aging, but if you can target all those things, that's fine, he said. But then he said, right, you're going to see if the hair is better and the skin is better, so he cares about, right? He cares about aging, and we're going to monitor that. But that's not what the FDA...
So
we're going to
which
the endocrine.
division?
We're assigned to the endocrine guys.